together, agree on the facts. 877-378-8
Institute. And today we're talking about Charles River, out on the Charles there, a floating wetland. And my guest today is Max Rome. And Max is a Ph.D. candidate at Northeastern University. Hello, Max.
Hello, Rob. How you doing?
I'm sorry to be indoors on the first day. It feels like the first day of summer, let alone spring.
Yeah, very exciting. Very exciting.
Well, I'm very excited about your little wetland that was set out in the Charles River last summer, and so let's get into that. And so you're a Cambridge. You grew up here in Cambridge, and
that's right.
Welcome to CCTV. So you're a natural here. So, what was your vision for the Charles River, or that led to this kind of project and stuff?
Well, I think there's a lot of ways to talk about this, but I think for me the vision is imagine a Charles River that is vibrantly healthy. A river that's filled with all of the kind of fish and insect species that we know could be here, and a river that is safe to go swimming in, safe to fish out of, a river where we're not having algal blooms every summer. And so much work is being done to help eliminate the pollution that's entering the river, but this project is all about what is the piece of that work that we can do with ecology. How can we kind of help the river improve its own ecology, give it kind of a step up to just be able to manage its own ecosystem a little bit better.
That is so cool because, for decades we've been working on closing down the effluent pipes and the runoff from coming in there, and redirecting the runoff to the right places and stuff. And this is like conservation 2.0. It's like, wait a second, we can do restoration work. We don't have to just turn off faucets and stuff. We can actually grow stuff or make stuff. So what are you doing?
Yeah, well, I just that is exactly right. And that's exactly how I'm thinking about it too. That, kind of from a generational perspective, there has been such incredible work and such incredible heavy lifting, since the mid 70s when the Clean Water Act was passed, to be like, okay, what is this pollution that we're pouring into the river? How do we stop doing it? How do we understand the risk associated with it? And that work, has been the work of, decades and decades. And now we're seeing kind of the fruit of all of that labor. But what we realize is that it hasn't gotten us all the way. All of this kind of pollution removal work has gotten us from, on the Charles River, from like a C or a D all the way up to a kind of a B+ or an A-. But if we really want this thriving, healthy river, there's something else that we need to do to kind of close that final distance, that kind of last mile. That's absolutely how I think about it, kind of as an environmental engineer. That, I mean, just as an editorial, from my perspective, I think environmental engineers have a lot to learn from the field of ecology in terms of understanding how ecological processes can actually be the solution to some pollution challenges. And that's not been something that we've been very good at, knowing how to model or design.
Well, it is exciting that the engineers are taking your path you're talking about, and they're calling it biomimicry, where we look at the way something natural works, like the way the squid changes its skin color.
Right.
Gee, can we design roofs to change their color with with black to white and stuff? So it's real. So you're kind of doing bioengineering. So tell us about your bioengineering process.
I've got my door banger, and if it does that again, I'll deal with it. But this is, I think biomimicry is an interesting kind of way to talk about this. What we are doing is similar to biomimicry in that, if you came to the Charles River 400 years ago, you would have seen this tidal estuary. You would have seen a place that was, say, 70% wetlands, 30% open water. And now what we have is all open water. And those of us who spend time on the Charles know that the river banks in a lot of places are kind of, vertical granite walls. And we have this urban water body that is different in so many ways from whatever it was that it used to be, that used to work, that used to function, that used to have a healthy ecology. So it's our job to kind of come back in and say, this river, unfortunately, it's not going to be restored to be a tidal estuary, but what's the kind of analogous environment? What's the kind of novel wetland habitat that we can create? And that's, where the floating wetland comes in. It's very difficult to imagine where you can put wetlands back into the lower Charles, just because it's so urbanized, it's so used, it's plays an important role for the city in receiving stormwater. So, people worry about flooding if you put stuff into the river. But a floating wetland is a way to kind of recreate some of those incredibly important things that wetland plants do, but in an urban context.
Yeah, and you don't have to displace any boat club or any rowing club or anyone who's got a piece of the water side,
Yeah, exactly. And hopefully, when we kind of look down the road a while, hopefully this could be something that, you could imagine kind of ribbons of floating wetland or places along the shoreline where there are docks and walkways out through the floating wetlands, and it could really, activate the river for boaters and kayakers and people who are fishing and people who are having a picnic. So, I think it's a great example of a project that can solve some problems, but also, create great opportunities for recreation and just enjoying this beautiful river that we all have.
So you just sawed off a bunch of marsh grass and floated it out there, and it's like a floating bog, or how do you deal with a floating wetland?
Yeah, so while the idea of a floating wetland is a kind of natural idea, the matrix itself is very much man-made. It's essentially a buoyant raft that has kind of this rigid, buoyant, framed out superstructure, and then within it there are planting rows. And very simply, the way it works is that you kind of put little wetland plugs into little tiny little 2-inch plants into these planting rows, and over the season they root out and they stabilize themselves on this wetland matrix. But the main power of this is that the roots go into the water column, and as they dangle down to the water column, they're sucking up nutrients. They're home for biofilms and all sorts of interesting bacteria and kind of tiny little animals, and that's what this project is all about.
And we're finding that when these marsh grasses or any kind of grass photosynthesizes, it draws a lot of carbon dioxide out of the air that it's then mixing with these the bacteria and fungi, helping it create nutrients for and pump out liquid carbon to feed the Charles River ecosystem, basically. I mean, what a great double-sided something.
No, that's a really good point, and to me a really interesting point, because there are some people who study these biofilms, and they say, like, kind of have an engineer's reductive view of these systems, and will say, well, plant roots are good, but we can just recreate kind of what happens with plant roots by just putting down a sheet of non-woven geotextile or something like that. But there's very interesting and very important kind of interactions between the plant roots and the biofilms. We're all just kind of starting to understand what some of those interactions are.
Are you talking about the rhyosphere?
Yeah. But what's interesting is in this kind of hydroponic environment, that rhizosphere is even weirder.
Yeah.
Yeah.
I mean, but we know more about the oceans than we know about the rhyosphere. And we don't know a hell of a lot about the ocean. And so you're combining the two, actually, I guess.
Yeah, it's really, really interesting. But I mean, our research is a little bit zoomed out from that. So,
yeah.
What we are looking at really specifically is the food chain in the Charles River. And as some of the folks who are watching this might know, one of the really big issues that happens in the river every summer is that there are these blooms of blue-green algae, of cyanobacteria. And cyanobacteria is, a really amazing organism. It's the organism that, invented oxygen, 2 or 3 billion years ago. And it's come up with a lot of great tricks for how to survive on this planet. But in the context of the Charles River, it's kind of a lowest common denominator type organism. It's the organism that's going to grow really quickly in the absence of a healthy connected food chain. And that's what we see. And as it grows and as it blooms, it does a lot of things that makes it harder for the river to support a healthier food chain, for it to support the type of fish that should be in the river, the type of microorganisms that should be in the river. And that's what, we're kind of hoping that this floating wetland can be an intervention to improve that dynamic.
Well, that's the number one priority. And I'm really glad you're onto it. I mean, we are really suffering from harmful algal blooms. And, cyanobacteria is one of them. It's not all the blooms are cyanobacteria, but that is definitely one of them. And the other ones, will also benefit from your wetland raft and stuff. There's it's gotten so bad that, the plants are blooming and eating up the oxygen and some secreting toxins to the point of creating ocean dead zones.
Yeah.
A situation where thousands of menhaden were chasing striped bass in Boston Harbor, and they went up the Mystic into an ocean dead zone, and they all rolled up dead. And there they were on the Everett shore there, right below Cisco Tire. Thousands.
Yeah, it happened last summer, right?
A couple summers ago. Yeah.
It's amazing.
Nutrients in the water was feeding the either cyanobacteria or algae, whatever it was, and causing that situation. So for you to create organisms that'll consume stuff so that we won't have so much, make up for all the nutrients that's pouring in there, that's really exciting. So what kind of organisms are you trying to foster and encourage?
Yeah, so our research is really focused on the zooplankton, which are kind of the link between algae and, small fish. So, zooplankton are these, herbivorous grazers. They're kind of like microscopic cows that are wandering around in the water, munching on algae all the time. And what tends to happen is that in a water body like the Charles River that's very turbid, that has a lot of algae and a lot of cyanobacterial blooms in it, you get only the much smaller zooplankton, kind of the lower order zooplankton. Rotifers are a really good example. There's tons of rotifers in the Charles River. And then, this is a bit of simplification, but, as you get to a healthier and healthier water body, you start to see kind of the higher classes of zooplankton, cladocerans, copepods, and in particular, a lot of people who study lakes like to look for, Daphnia, which are sometimes called water fleas, but they're really, really cool little organisms that are very, very good at eating algae, but also are very delicious if you're a fish. So, so what essentially what happens is in the Charles River, there is so little refuge for these larger zooplankton that if you're a fish, you're going to eat them right away. You know, they don't have a chance to kind of hide out on a lily pad stalk or hide out in the reeds. And you know, the fish kind of gobble them up.
Well, that's really cool because you see the when you walk along the Esplanade, you'll see little bits of herring.
Yeah.
There and bread, but you know, the little fry that are come out of the eggs, they're going to be hungry guys, and
yes,
and we're providing food for them.
Yeah, that's exactly right. I mean, and that's another, I think, piece of the story that I like to think about, you know, is that there have recently been some headlines about what's happening with migratory fish. You know, the kind of headline is catastrophic decline in migratory fish, that, you know, since the 50s, it's possible that there are 70% less migrating fish than there were in 1950. And, you know, a lot of that has to do with dams. A lot of that has to do with pollution, but a ton of that has to do with, you know, wetland destruction, which was, you know, kind of the nursery grounds for all of these fish. And that's another, you know, that's another kind of piece of what we hope these wetlands can help support.
Absolutely, because you've got to have functioning ecosystems wherever the fish may be, you know, and in their natal years or in the rivers. And so, you know, the wetland is a great way to go. I mean, you've got the whole thing going.
Yeah. I mean, and it's just so amazing to me to kind of spend time on the Charles River and just realize that, you know, wow, the health of this river is, you know, connected to this river herring population, to these alewife that then go out and kind of support this other incredible ecosystem, you know, all the way up to, you know, humpback whales or, you know, seals or any amazing creature that you can think of.
Yeah, I appreciate it because at the Ocean River Institute, we've been working with the fishing industries to make sure that they take an ecosystem-based approach, meaning that the herring are forage fish, and
yes,
you've got to leave them for the other critters. And so we sued them in the court, and they now have to take an ecosystem-based approach. But there's no point in saving them if they can't survive in the rivers because there's no wetlands there and stuff. So we need, everyone is working together, you know, and then we managed to get them to move the herring fisheries further offshore so that they'll catch less of the river herring and just try to do the Atlantic sea herring. Because one trawl can pull 800,000 tons of fish, and that's a year class coming out of the Charles River in just one pull. So we want that fishing offshore, you know. But yeah, so do you want to show? Should we look at some pictures of your floating lab and stuff?
Yeah, I can show some images of it out on the river. Let me just take a second to make my technology work correctly.
I should have said that before I gave you the whole spiel about herring. So cool. They've got little oar feet sticking out, and then the Daphnia. You know, they raise Daphnia for in aquariums, and I've had them in the classroom and stuff. They're really easy to keep and stuff. But
there's, I mean, such amazing creatures like that. You know, they have blood cells. They can become intoxicated if they're exposed to alcohol. They have this really interesting life cycle where, you know, most of the time all of the Daphnia are female and they give live birth, but every once in a while at the end of the season they make some male, procreate, and then they, you know, lay these winter eggs. So it's just so many cool little, you know, microcosms in the river. But here's a look at the wetland last summer out on the river, kind of attached to the buoy. It's out near the Salt and Pepper Bridge, kind of between there and the Museum of Science.
Right, I can see Mass Ioneer behind it there.
Yeah, exactly. And you know, here's another shot of it. I like this picture because you can kind of see the seawall behind it, and then you can see the floating island, and just you know, it's a dramatic reminder of what's missing on the river. You know, and I think we get so used to what's there that sometimes we don't realize what's missing, all this vegetation. Yeah, this is a shot on the wetland where you can see the marshmallow, which is a New England hibiscus that's blooming. We planted about 19 or 20 native New England wetland species, and this was one that really did well on the island.
That's cool. Marshmallow has all kinds of medicinal uses too and stuff, so that you could have no trouble finding people who would like that plant at the end of the year or something.
Oh my God, and it's such a beautiful plant, and the insects love it too. I mean, it's good for everybody. And this is, so next I've got kind of a couple research images. So this is an image of the summer bloom. So this is a cyanobacteria called a phanezomonin that builds up into these very long rafted filaments. And this is it kind of concentrated in our plankton net. This is what it looks like at like 100x magnification. So you can see in this image, there are actually the 3 different cyanobacteria that are most common on the Charles: Microcystis, phanezomonin, and Anabina, which is this kind of like pearl necklace looking cyanobacteria. And I mean, cyanobacteria are another, really amazing, fascinating organism because they are single-celled organisms, but they're colonial, and they do these really unique things. Like they can tell one cell, okay, don't produce any chlorophyll. Instead, you're going to fix nitrogen from the atmosphere. You're going to pull nitrogen out of the atmosphere so that we can fertilize ourselves. And then here's some shots of kind of what our summer research looks like. These are some of our great summer research assistants. And every day we go out there, we net zooplankton, and we, count them to see how abundant they are, and then we measure them to see how large they are. So that's kind of where the rubber meets the road with some of this work. And this is
back. So that's a net that they let in the water, and then as they pull it up, it all goes into the cup at the cod end there, and then you're going to show us the critters that you catch.
Yeah, that's exactly right. So you're filtering all this water so that you can concentrate the zooplankton down in a little volume where you can actually count them. And this is a picture of a nice adult copepod with a really nice clutch of eggs that was taken from the wetland. And this is exactly what we hope to kind of see more of at the wetlands, because what these guys do is they just basically eat algae all day long.
Yeah. Marvelous.
Yeah.
Yeah. So we've just got a couple of minutes left, and it's just fabulous what you've been doing. How can people learn more about the Charles Floating Wetland?
Well, we're very interested in connecting with people who are interested in this project. So we have a research website at charlesfloatingwetland.com. And the Charles River Conservancy is our partner with Northeastern on this project. And you can find their website at thecharles.org. But we are going to be running some kayak tours this summer. And there's going to be a number of different opportunities to, learn more about the wetland. We're going to be planning the second phase, kind of a wetland expansion that'll happen in, hopefully in the next few years. And we're really interested in engaging with, the communities in Cambridge and Boston to kind of hear people's ideas, hear people's concerns, and kind of design a project that is really going to be exciting and relevant to everybody.
Well, I'm looking forward to going on, copepod safari with you and stuff.
Yes. Oh my God, it really is a copepod safari. So exciting to see them underneath the microscope.
Maybe we can, get up some kind of live cam going or something.
Yeah.
So spread out that you can't really, put an underwater camera to catch them because you need to run them through a net and stuff.
Yes, they're far away from each other and they move very quickly.
Well, we have at the Mystic River Dam, we have a fish counter. So maybe we could install a micro-sized little, straw-size copepod counter that the water flows through.
Yeah, that is an amazing idea. I have a colleague at Northeastern who is working on a flow through microscope like that. You know, that's exactly the concept, that it could kind of image something that's passing by the camera, and you'd be able to do
eating copepods. You know.
Yeah.
Well, Max, Rome, thank you for taking time to talk about the Charles River Wetland, floating wet lab.
Well, thank you so much. I really appreciate the opportunity, and I hope that you and I can meet up out on the river sometime this summer, maybe.
Yes, and for all of you listening, thanks a lot. Please take care of yourselves, and then take some care of this planet of ours. Thanks for listening. So the machine automatically shuts down. Maybe it'll get our little banter here, but oh yeah, no, it's gonna be fun. I gotta get out on the river sometime.
Yeah, it's really, the one really nice thing about it is it's really close to the Kendall Square canoe and kayak rental. So it's great to get out. All right, well, thank you so much, Rob.
Yeah, hold on a sec. Because Steph will come on and then we'll really off and stuff. But usually it comes out, yeah, it should be over by now. But yeah, I did my student teaching at the Tobin School.